Adrienne from. So Adrienne is leading the plant virology team at FERA Science Limited. So he's doing regulatory diagnostic, but also the group is doing research for novel diagnostic techniques, and of course it includes application for high throughput sequencing. So Adrienne, floor is yours. You didn't mention in your bio your talent as a teacher, unlike Bart.
I don't like to boast. Unlike that.
Yeah.
Okay. Good afternoon. I recognise a lot of faces, but for those of you that don't know me, I will start with an apology. My sense of humour is my sense of humour. What comes out in the next 20, 30 minutes, I've got no control over, so I'll apologise now. So I come from FERA Science Limited in the UK. Back in the days that we were still called Central Science Laboratories, or CSL, we were among the first groups in the world to publish on the use of high throughput sequencing in plant health diagnostics. And that was back in 2009. Since then, a lot of our development work has been about how do we get HTS into routine diagnostics. Yes, we use it in research. A lot of the things I'll talk about here will apply to the outputs of research work in the same way they will apply to routine diagnostics. I suppose we need to define what do we mean by routine. We're still not at a point where every sample that comes into virology goes into HTS. There's no point. If it's a potato sample with a mottle, why would you HTS it? It's probably PVY. Let's test it for PVY by ELISA and get it out the door quickly. However, there are things that come in that the conventional diagnostics don't quite make sense. We flip to HTS. There are also things that come in that we have no a priori knowledge of what may be in that plant species. So at that point, do you do a suite of ELISAs, PCRs, real-time PCRs, a bioassay, possibly use an electron microscope, by which time you've probably spent 6,000 or 7,000 euros on your diagnosis? Or do you do it on the high throughput sequencer, where we're currently turning them around for about 500 euros per sample? I know what the sensible way to go is. If you put it on a high throughput sequencer, you at least get a definitive answer in a controlled amount of time for a controlled cost. And for that reason, a lot of our work has been on how do we get to a point we can use the sequencing for routine diagnostics, but more importantly, with our regulatory hat on, how do we stop the risk assessors and risk managers panicking every time we knock on the door and say, we've got a new thing? Because that new thing's exciting for me as a virologist. That's what I get out of bed for. I mean, how many people can say that they found a new species? Well, okay, by the time you're onto your 200th or 300th new species, you wish that you weren't finding new species, because that just gives you a headache. But that's why we get out of bed. But if you're on the risk assessment side and the risk management side, that's a headache. So I'd say a lot of the work we've been doing is about how do we ease some of those nerves. I am a virologist. A lot of the examples I give will be virological. I make no apologies for that, unlike my sense of humour. But virologists are early technology adopters. Because of the nature of the pathogen that we deal with, we have to have technology to help us diagnose what's wrong with it. So our discipline is about 180 years old. If we go back far enough, we start with a Chamberlain filter, running a liquid sample through, and if it passes through, it's a virus. If it doesn't pass through, it's not a virus. We've advanced a little bit from there. We've gone through electron microscopy, bioassay, ELISA, conventional PCR. We've done in-field diagnostics. So this is our in-field diagnostic development pipeline at the top. Yeah, we've gone to higher and higher throughput for molecular diagnostics. And at the end, we come out at HTS. I will add, that's a very small sequencer, not a very large dog. So, the interesting thing for me when I look at this development pipeline is you can split the diagnostics we use into targeted and non-targeted methods. So by targeted, I have a specific thing I'm looking for, I go out, I test, say, is that there? And I get a binary yes or no answer. So again, analyzer for PVY, I've got PVY, everyone's happy. The non-targeted diagnostics are, I'm not quite sure what I'm putting into the system gives me an answer, I've then got to make sense of that answer. Well, that's what electron microscopy does, that's what bioassay does. To a certain extent, that's what genus level PCR does. Yeah, when René van der Vlugt published on genus level PCR for potyviruses, we didn't have the panic that we now have about what comes out of the high throughput sequencer. And in principle, all a high throughput sequencer is giving you in terms of results are multiple genus level PCRs that you then have to interpret what that result means. You know, from PCR, we've had bioinformaticians who need to look at the Sanger sequencing data and tell us what it actually means. All we're doing here is doing that in a massively parallel way. And so, as I say, a lot of what we've done is about how we can ease some of the nerves around what's coming out of the system. And if you think about how we report actions, so we found the thing, what do we do about it? So we found the thing, whatever that thing may be, usually a novel virus in our case. You know, you have all of these questions to come through. Do I need to confirm it? Is it regulated? Is it novel? So if it's novel, it's not going to be regulated. So then how do I know whether I need to panic or not? If it's an import sample, is it present in your territory? Yeah, are you taking action on something that may already be there and widespread? You just didn't know it was there because you didn't know to look for it because it's novel. So you may be unnecessarily taking action. Is it a known host for that pathogen or is it a new host? If it is present, is it already widespread or is it a single sample? Will it impact this crop, other crops, or the environment? And I think Bart used the C word already, context. Context is absolutely crucial in making sense of what comes out of the sequencer. You may have something like this here. So this is a glasshouse with brown ghost fruit virus, maybe spreading massively, 70% of the plants are dying. Or you may have something like this in a carrot field, single yellow leaf in the middle of nowhere. Is that something I should worry about or not? Because that was a novel virus. So is that something we should panic about? So you've got all of these questions. Do you have that data? How do you generate that data? How long will it take to generate that data? And given that it could take 3, 4, 5 years, I think with this carrot one, we're now on about year 14 of the research arc to try and understand what that's doing, how big an impact it has, what's transmitting it, how do we stop it? Are you happy to live for 14 years before you take action? At some point you need to take action. And therefore, you're taking action with a degree of uncertainty about what you're taking action on. And then you come down to your risk appetite. And even within the constraints that we currently have, we see that some countries have a greater risk appetite than others in terms of when do you take action, when do you not take action. And all I want to do today is start to introduce some of the discussion points for this afternoon and to get people thinking about all of these questions and at what point should we be taking regulatory action. But the other thing to remember is these are just general diagnostic questions. This isn't special for HTS, but because HTS does that generic PCR or that bioassay in a massively parallel way, what it does is it brings these questions sharply into focus and we now have to deal with them. I'll just take my word for what I'm saying here. There's a really good EPPO document, PM 751, and in there it talks about confirmation and biological interpretation and reporting. So is it just critical cases? Is it previously uncharacterized pest? How do you deal with that? How do you interpret the biological relevance of the identified targets? Is this a known plant pathogen? Is it just bycatch? Something, and what I mean by bycatch is a term from the fishing industry. You went out, you didn't mean to catch that, but you caught it by accident. Have you just picked it up without knowing it's there? What information do you actually need to support your decision, and how much depth of that information do you need? And on reporting, do you report everything? And we'll talk about some situations in a moment where we've got real data where I would ask the question, do I report everything? If it's not a plant pathogen, do I report it to the animal health authorities or the human health authorities? When do we panic? So I'd say we've been working on this since 2009, and we moved to what we call our routine diagnostics for the Plant Health Authority in about 2011, 2012. We then got to a point where we were doing what we call a routine run around every 6 to 8 weeks. We're now on run about 53 or 54 of those routine runs, but interspersed in there we also have research and development runs going on, so you've probably got double the number or triple the number of research runs. And all of them produce this kind of information. And so by this point, we've decided on what platform we're using, we've got our bioinformatics pipeline nailed, we're running our controls, everything that you heard about this morning, we're quite happy with where we're at. We don't have accreditation, talk to our accreditation authority about that, don't talk to me, but we've got some validation data. As I say, we're happy with what's coming out of the system, but what we need to do is try and make sense of it. But obviously, if you just sent that to the risk management team, they would go into meltdown. If you send them that every 6 weeks, you'd never hear from them again. So what you need to do is you need to take them by the hand and lead them through. I'm a subject matter expert. I know viruses. What I can do is help to interpret this data for the risk managers and to help lead them through. And what I thought I'd do is, this is actually data from a real run a couple of years ago, and I just picked it at random from one of those 53 runs that we've done. And I just wanted to give you a few editor's highlights of the kind of thing that comes out of the system. So these are just a selection of that stuff that was on the previous slide. Everything we do a de novo assembly, we blast end it, and once we blast end it, we then blast exit as well, so that we've got the transform sequence, and then we can reality check what we've got. And here you've got this Solanum macrocarpum, which we'd found Tamarella fruit ring virus, some relatively novel potty. It's not novel, it is on GenBank, but it's not formally reported yet. And we found it within this plant, so using the generic pottyvirus PCRs of all things. And so we went back, we HTS'd it to try and get a full genome for confirmation. What we also found out was there was a second novel pottyvirus in there as well. But then we got these 4 fragments of Nerine virus X. And if you think back to what Sebastian was saying about the leakage in the runs, well, actually, we know that we had a couple of Nerine virus X isolates actually on this run as well. And that's just leakage being picked up from there. We also got these 4 fragments, quite significant fragments of Begomovirus. So what do we do about that? Well, actually, in terms of the context, that's an import sample. The fact that we've got a novel pottyvirus and pottyvirus that's non-indigenous to the UK in there means we can take action and hold it, stop it, turn it around, whatever. At this point, we don't need to worry about the Begomovirus. We're still working on it. But what it means is we can take action quickly, and then 2 years later, I think, is this 2021, 3 years later, we're still working out what that Begomovirus is. So we've got this capsicum sample with a large fragment of a ranunculus white mottle. Well, actually, that's because that capsicum sample had gone through all our targeted testing. We hadn't come up with anything that we could think it was, but it had symptoms. We wanted to know what it was. So we put it on the sequencer, and it shows ranunculus white mottle. It's a UK glasshouse sample. It's been reported to DEFRA for action, and there's a new host and country record. And that was simply, I think the action was just to remove the plants and some surrounding plants from the glasshouse. We didn't confirm that ranunculus white mottle. Workers, with a bacterial virus, a Parpiti virus, that's an endogenous plant virus, and some avian virus. Well, there's nothing in there that would worry me as a plant virologist. UK field sample, no plant health interest. Move on. We've got enough to worry about. We're still trying to work out what that Begomovirus is. Again, capsicum annum. We've got, again, these fragments of Nerine virus X. We've got some Chenopodium quinoa mitovirus. That's probably a virus from, I suspect it's not a plant virus, but anyway, but it's an endogenous virus. We've got fragments of herpes virus. Sometimes it's better off not to know what your team have got. And we've got 17 fragments of this avian virus as well. But in the BLAST X, we've got tobacco vein clearing virus. And again, UK glass house sample, no plant health interest, move on with your life. And finally, we've got these 2 marine virus X samples, and these were actually part of the Fresco project, where we were looking for historical isolates to try and make sense of what's on GenBank and give some actual depth of understanding about the different sequences on GenBank and the viruses that don't have any supporting sequence at all. Marine virus X was one that didn't have any supporting sequence, so we were generating novel sequence for this. Again, no plant health interest apart from the fact that we're supporting whatever's on GenBank. So we want to be alive. But HTS reporting for plant health has been covered before, and I'll draw your attention to this paper from Sebastian from 2017. This has been renewed under the Inextivir project, and I think, is that about to go live? Couple of weeks. I never understand how Frontiers works, but yeah, it's about to go live. And basically what we have to think about is that plant health regulations are heavily based on species lists, and high throughput sequencing chucks up lots of novel stuff. So the species lists don't really work in that respect. Essential questions for PRA, I'll draw you back to that original slide: presence, incidence, distribution, and impact. And as I say, how to handle this increase in novel pathogens was raised by Sebastian in this document in 2017 that's now been revised. But how to handle these have real implications for us as plant health scientists. And those implications are time, cost, because yeah, that 14 years of research work on that novel carrot virus has probably cost about £2 to £3 million, mostly of industry money, but we're still chasing it. You don't have that money to do every single virus that comes out of the sequencer. And you've also got your skills and resources question. A lot of those traditional diagnostic skills aren't cheap, they're falling out of use. I was just talking to Annalene earlier about DODA transmission, and that's something that we don't even have DODA going. We're going to pop over to see Annalene and see if we can work out how to get that within virology at FERA. But again, all of those traditional skills that we relied on and were developed for years have now fallen out of use because of the switch to molecular biology. And more importantly, how do you prioritize what you work on? If you're faced with a dozen novel things from every single sequencing run, where do you even start? And I'll be honest, most of the time we're starting on what's easy to work with. So the contact transmitted stuff, the tabamo viruses, things like that. The non-persistently transmitted insect viruses may get a look in. If it's something like a Polaro-virus that needs a lot more work and a lot more technical skill to play with, that sits further back down the prioritization line, unless we think it's worth panicking. So just to think about how much confirmation we need, and this is really kind of one extreme of the example. So this was our EUCAS work and using HTS to confirm virus identification. Inspector went out, found some EUCAS plants, we did some ELISA. We agreed categorically we wouldn't do HTS on this, because if you're testing plants that you don't have any clue what you might find in them, when you start finding novel things, it just causes confusion. So we only screened using conventional diagnostics for quarantine pathogens. That screen suggested that we had Andean potato latent virus, quarantine virus, but we couldn't confirm it with conventional diagnostics. We flipped to HTS, and what HTS showed us was actually we had 2 novel thymoviruses that sits in between 2 regulated thymoviruses, and we had to make a call: do we think this is a regulatory risk or not? Well, everything north of that purple line is a solanaceous affecting thymovirus from South America. So chances are they're solanaceous infected thymoviruses from South America. They don't belong in the southeast of England. We felt we were within our rights to take action on that. And this then led to EUCAS being included on the European legislation as a high-risk plant species. And I like to think that was our parting gift to the EU just before we left. Sorry, Francois. But to break down, and I'd say this is kind of the extreme example of taking confirmatory action. This was our flow chart for the action we took. So we got samples in, we did our initial diagnostic. When we found APLD, we notified the NPPO, but we couldn't confirm that, so we switched to high throughput sequencing. That then showed why the conventional assays we were trying to use didn't work, but we then flipped again and we confirmed our confirmation testing as well. So even though our HTS was confirming our original conventional diagnostics, we went back into a confirmation step before we actually took action in the end. And I say, you can confirm until the cows come home. At some point, you have to decide if you're taking action. But with this one, we had that 3 levels of confirmation. You need to think about its confirmation necessary. So PM7151 and EPPO principles, we confirm critical cases with a secondary method, preferably using a different biological principle. But what does that actually mean in practice? Yeah, we take that approach in regulatory work, but also for publication requirements. So if you've done an antibody-based test, then a molecular test is a different biological principle. If you've got a biological test, then a serological or a molecular test is a different biological principle. However, if you've done a qPCR, is a conventional PCR a different biological principle or not? If you've done a conventional PCR, your Sanger sequencing isn't a different biological principle, and it's actually codependent on your initial test anyway. But some people will take that as a confirmation. So what questions we then had is, can you build risk assessment data and confirmation into your general workflow? But also there's a question about, can sample context plus test validation for HTS allow you adequate confidence in your result to then take action? And this is an example of a piece of work done by Amy Fowkes in my lab, where we were looking at P as a way of developing a method for doing generic survey work of a crop, where we actually built a confirmation step into our workflow. So we did 60 sites over 3 years. From each site, used this 100 metre by 100 metre grid. We took about 120 individual leaf samples. From each field, we bulked all 120 leaves, and we put those through MySeq, but also we subsampled from that 120 individual leaves and did smaller bulks that we could then confirm anything that came out of the high throughput sequencing, we could confirm using conventional methods as well. And doing that, we got the cost of sequencing down to less than €2 per leaf that we tested. What we also did, we reran this at a greater depth and showed that we hadn't missed anything. But the outcomes of this were that we managed to get presence and incidence of viruses based on our original HTS sequencing plus the confirmation testing, and we showed that turnip yellow virus was actually the most prevalent virus, first time it had been found in UK fields. It was showing no visible symptoms, so we've also got to have a question about what we mean by impact, because it doesn't show visible symptoms, but it does reduce yield by about 40%. So you've then got to think about further questions within your impact assessment. It also gives the first UK records of several viruses, but ultimately these weren't making an impact that we could see, and there was no plant health action taken. At the other end of the confirmation scale is this example that I got from Marley and Bottomans, where again, context is king in terms of being able to make your decision on should you take action. So regulatory survey of sweet potato using the accredited method that Bart presented. 4 different species were detected within that sweet potato. One's listed as an EU quarantine pest with nearly complete genome found, so sweet potato chlorotic stone. You found a sweet potato regulatory virus in sweet potato. It's coming up at depth. You've got nearly a whole genome. Can you take action without confirming it? Do you really need to confirm it, or are you happy that it probably is what you think it is and you can take action? So, just to finish off, I want to talk about some of those problems that we've got. So, here we're thinking about that problem of how do we prioritise and the gap between what we're finding and what we're able to characterise and work out the risk of. And Sebastian produced this paper on this emerging biological desert. That's different to this. This is biological desert. That's different. But what that paper showed is that of the new publications that are coming out on fruit tree viruses, the majority of them have got a nearly complete genome and a primer designed for further diagnostics. But less than 50% of them have any kind of risk assessment data available, whether that's survey work, biological characterization, and so on. And so we have this growing biological desert when you think that we're actually increasing the number of known viruses by about 10% per year in terms of what's on the ICTV, and that gap is ever widening. The other thing to think about, and this is increasingly becoming an issue with things like virome studies where we're going out into the wider environment and picking stuff, is what you're finding in the sample or on the sample. Is it actually infecting, or is it just, as I said, bycatch that you've picked up? And the examples I've got here are actually from a brown dust fruit virus infected glasshouse. None of these samples have actually been anywhere near a plant. They're all just swabbing of different surfaces. You'll see here, this is my favourite, the Tyvek suit. It's come up with a CT of 16. Well, that's actually Anna, who works in my lab. She went up on the scissor lift to swab the roof of the glasshouse. When she came back down, she had a rusty mark across the back of the Tyvek suit. We swabbed the rusty mark, and that's come up with a CT of 16. That's not a plant, but if I tested that plant, I'd say it was positive. So you've got to think about, does this make sense? And actually, is there a biological context? Now, if you're just looking for is it present, that may not matter. If you want to take plant health action, it probably does. And so, to go back to that original slide, you've just found this thing, and these are the things I want to think about this afternoon. Do you need to confirm? Is it regulated? Is it novel? Is it present? If it's present, is it widespread? Will it impact this crop? How do I know if it will impact other crops? Do you have these data? Well, the answer to that is probably not. What you need to think about is how much data do you need before you're willing to take action? Thank you.
Thanks a lot, Adrian, for all these questions which speak to us because, in fact, in the job in EPPO, we are both doing the diagnostic part and at the same time the risk analysis part where you really have to scratch your head. Am I going to do anything about this? So you've raised very important questions which are more prevalent with HTS and it's everything. And the last graph you show is really, at the same time we see that all the recommendations that do not stop after just producing a new sequence, but go further. Well, how many percent?
Less than 50. Yeah, 90% have a genome and then
and
less than 50 have any biological. I think less than 30, isn't it, have any real biological.
But it's really great to see that some groups are really pushing for having this information, but I shouldn't talk too much. Are there any question for Adrian, or there will be lots of discussion this afternoon in the session on reporting, but well, it depends if you think it's important for the group. I think the next speaker is connected, but let's take the moment to have a question.
Again, on this confirmation, there are also cases in which you repeated the sequencing, so not using enough PCR, but you repeat the sequencing, or you can still then go for ELISA or PCR and so on.
And we do a similar thing that we'll switch it around. We've also done followed Illumina with MinION sequencing as well, just to try and get that second result.
Thank you. There is also oasis in the desert, because if you look, for example, the tomato brown ghost fruit virus, it has been completely characterized in very short time because it was really a big threat, and we have it recently for another tomato virus that is causing an emerging problem. So in fact, even if the desert is also for the viruses that does not seem to be really epidemics or cause high symptoms, but globally, the community is going where is, I think, quite efficiently using the small resources they have because they see the threat, they see the growing problem, and so there's oasis in the desert.
You're such an optimist. I've never had you down as an optimist.
Okay, thank you, Adrian.